Welcome to Session 2 of the Australian Academy of Science Fenner Conference on the Environment. In this session, we will hear more about the cryopreservation of exceptional species. Please use the question and answer button at the bottom of your screen to submit questions, noting which speaker you would like to ask, as well as your name and organisation. Our introductory speaker is Dr. Valerie Pence, the Director of Plant Research at the Centre for Conservation and Research of Endangered Wildlife at Cincinnati Zoo and Botanic Garden. Valerie is going to talk about her long-term cryopreservation research, as well as the Global Exceptional Plant Conservation Network. Then, Dr. Bryn Funnicotter, research scientist at Curtin University and Kings Park, will introduce us to cryopreservation in Australia. Dr. Jayanthi Nadarajan, team leader of germplasm conservation at Plant and Food New Zealand, will present her work on the cryopreservation of embryonic axes. And Dr. Daniel Ballesteros of the Millennium Seed Bank will introduce us to the science of fern spore conservation. Enjoy these presentations.
Good afternoon, everyone. I'd like to thank the organizers of this Fenner conference for the invitation to talk today about exceptional plants and the network that is needed for their conservation. As you all are aware, exceptional plants are those that can't be banked effectively long-term in conventional seed banks. And you've heard today how these will require methods beyond conventional seed banking and will pose a significant challenge. As you've also heard today, one approach to this challenge will be living collections. The other approach will be to use cryobiotechnologies to conserve seeds or tissues long-term in liquid nitrogen, which is what I'll be focusing on today. Within the approach of cryobiotechnologies, a variety of tissues can be targeted for cryostorage, but not all methods are equal in terms of their efficiency and use of resources, in other words, their cost. They can be looked at in terms of their use of resources compared with conventional seed banking. Short-lived seeds and spores can be handled in much the same way as for conventional seed banking, but using cryopreservation in place of -20 degrees centigrade. Tissues such as isolated embryos and dormant buds, however, will generally need some additional recovery methods, such as in vitro growth or grafting. Shoot tips and somatic embryos require in vitro initiation to produce the propagules for banking, as well as to recover and grow them after cryostorage. As one moves from seed-based tissues to clonal tissues, there is less genetic diversity captured per banking, while at the same time, there is an increase in the time and resources needed for these methods. Despite that, once any of these tissue types is banked, it can be maintained long-term in cryostorage with little additional input of resources. The choice of method will depend on what is possible with the species and which methods will be most effective for the least cost. Cryopreservation has been shown to be a safe and effective method for conserving viability at least over the 2 to 3 decades that the methods have been in use. In a study from our laboratory, a variety of tissue types and species were removed from liquid nitrogen storage after from up to 23 years in storage. I want to acknowledge Dr. Danny Ballesteros, pictured here, who did much of the recovery evaluation of our cryopreserved collections. Short-lived seeds of salix and populus were shown to have much better survival in liquid nitrogen over that time than at -20. Similar results were seen with fern spores of several species, pollen of Castanea dentata, and embryos of Juglans nigra. Shoot tips of a variety of species also showed good survival after long-term cryopreservation. One example is Hedioma tudsinii, a federally endangered species from the mountains of New Mexico in the southwestern United States. It's a species that's no longer making seeds, making it exceptional, but we have initiated multiple genotypes into culture and cryopreserved the shoot tips from those cultures. These results show the average survivals at the time the tissues were banked using 2 methods, encapsulation vitrification, or EV, and encapsulation dehydration, ED, the 2 bars on the left of this graph. After 13 years in liquid nitrogen storage, there was no loss in average viability with either method. And you can see the later bars on the right of the graph. There were survivals between 30 and 40%, both before and after storage. This graph also shows that a newer method, droplet vitrification, gave much better survivals than either encapsulation dehydration or vitrification, illustrating that cryopreservation methods can improve over time. So the methods are available and proven effective, but implementing them on a scale needed requires a range of expertise and institutional commitments. In a very rough way, the process of meeting the challenge of exceptional species conservation will have 3 steps or groups of steps. First, we need to know what species are exceptional, which species are of conservation priority, and which conservation methods will likely be the most efficient and effective. There also needs to be information on the location of the plants and ways to access the seeds or tissues needed. This step may require input from taxonomists, field botanists, and seed biologists. Once seeds or tissues are obtained, the second stage will involve developing the protocols needed for effectively cryopreserving the tissues. This can be fairly straightforward in the case of short-lived seeds, or it may require developing in vitro recovery methods for embryos, grafting methods for dormant buds, or producing in vitro tissues for shoot tip or somatic embryo cryopreservation. All such tissues need to be tested through cryopreservation to document successful survival and recovery. So this step may involve seed biologists, in vitro, and cryobiologists. And developing a protocol for recovery includes having methods available for the growth of the surviving tissues into healthy plants, requiring horticultural expertise as well. Protocol development is likely to be the most expensive step in the process and is unpredictable in the length of time it will take. However, once protocols are developed, cryobanking can begin. This will involve input from collectors, and ideally from genetics, to help guide the collecting of the materials most important for conserving. Expertise in implementing cryo, in vitro, and horticultural protocols will be needed as well. For this overall process, a variety of institutions will be needed, including botanical gardens, arboreta, associated living collections networks, and governmental agencies. These can all be important for information and collecting, as well as sources of material. Those same groups, as well as academia, can play a role in protocol development. Finally, institutions with cryobanking infrastructure will be needed for the final step of banking and maintaining the collections. These cryobanks might be in the same institutions where the protocol development occurs, but they don't need to be. Labs without cryostorage capacity can contribute to seed testing and developing in vitro protocols. If they have access to liquid nitrogen, they can also develop cryopreservation protocols even if they don't have long-term storage facilities. These types of collaborations could greatly increase the numbers of exceptional plants that are conserved. While these steps are similar to those involved with conventional seed banking, the question is, can exceptional plant banking meet the efficiencies of seed banking? It's possible that it could for short-lived seeds, spores, and pollen, but it will be more difficult with other types of tissues. Thus, there's a great need for innovations in making the processes more streamlined and more broadly applicable. The newer cryo procedure of droplet vitrification, for example, takes less time to execute and seems to be more broadly applicable than some previous cryopreservation procedures. Finding ways to generate buds from tissues and cryopreserving those takes less time and tissue preparation than isolating shoot tips for some species. This is similar to the green globular bodies that can be generated from ferns, which also have survived well through cryopreservation. And more basic research into cryobiology and in vitro biology is needed to improve our understanding of freezing stress and plant growth in vitro. It will likely also be more efficient for centers of expertise to develop around tissue-focused methods rather than species, since methodologies are often similar across species. And having multiple centers worldwide to conserve in-country species could provide more ready access to tissues, more local knowledge of plant locations, etc. While innovations and approaches may not ever create equivalence with seed banking, more basic research and innovation is needed to help make these protocols more efficient and thus less costly. In order to focus attention on and facilitate the research connections and collaborations needed for the conservation of exceptional plant species, we're developing a website for an exceptional plant conservation network. It can be found at cincinnatizoo.org/EPCN. It will host an exceptional status list, information and links to information on exceptional plant research resources, and a directory of those who are working on exceptional plants in some capacity or have some expertise and/or infrastructure and would like to work with exceptional plants. This is the current first page of the site, which will be changed and updated periodically. To contribute to the information on exceptional plants, we have created the first iteration of an exceptional status list by evaluating over 23,000 species using data from the Seed Information Database and other sources. The resulting status list contains information on the exceptional status of a species, that is, whether it is exceptional, non-exceptional, or is lacking enough information to make a decision. If the species is exceptional, the list will indicate what factor makes it exceptional, such as lack of seed, desiccation sensitivity, etc. Currently, the list identifies 775 exceptional species, and approximately 1/4 to 1/3 of them are threatened. These should be prioritized for conservation action. We have also created a list of threatened congeners of exceptional species, which should be also of high priority. However, 775 is estimated to be 2% or less of the total number of species that are predicted to be exceptional. Thus, much more information is needed to identify the remaining species. In addition, information is needed to clarify species for which there is insufficient data. We encourage people with information on additional exceptional species or on species with insufficient data to contribute that information to the list using an online form that is also on the EPCN website. In addition to the list, there will be information on resources for developing protocols for exceptional plant conservation. There are a number of such resources available online, including the Australian Network's Germplasm Guidelines, as well as information from the CPC, the Millennium Seed Bank, and other resources. We will have links to these resources as well as additional information. An important part of the network is the opportunity to learn about and connect with other researchers, to learn from each other, to avoid duplication of effort, and form collaborations to more efficiently work on exceptional plants. Thus, the website will also include a directory. We encourage anyone working in any of these areas that support exceptional plant conservation to fill out an online form and add your information to the directory, which will be posted on the site. We also encourage researchers who have expertise or infrastructure that they would like to use to support exceptional plant conservation to also list themselves in the directory. In summary, cryopreservation is a safe and effective method for conservation, and there is a need for a variety of types of expertise and infrastructure to address the challenges of information and collection, protocol developments, and banking of exceptional plants. And hopefully the Exceptional Plant Conservation Network can play a role in facilitating that work. Meetings like this Fenner conference are particularly important in working toward that goal. I'll close by mentioning that we will be holding a symposium workshop in October on exceptional plant conservation as well. It is free and virtual, and it will focus on exceptional plant conservation and cryopreservation with a focus on oaks as well as other exceptional species. The talks will be pre-recorded and available ahead of time, and for those in Australasia and the Pacific, we will hold one shorter session at a time during which you can all tune in live. Registration and schedule information is on the EPCN website as well. Thank you for your attention, and thanks to the Institute of Museum and Library Services for funding this project. And I'd like to acknowledge the co-authors who worked on the exceptional list: Emily Beckman, Abby Meyer, Hugh Pritchard, Murphy Westwood, Joachim Gretzfield, and Jean Linsky. And thanks to our web manager, Angela Hatke. I'm sorry I can't be with you in real time, but I'm happy to answer any questions that are sent to me. Thank you.
Welcome, my name is Bryn Funakotta. I'm a research scientist at Curtin University in Kings Park Science, and today I'm going to give a bit of overview of cryopreservation in Australia, and then focus on shoot tip cryopreservation. So cryopreservation is an important conservation tool and provides a very long-term storage option, many species being successfully stored for more than 25 years, and some species even more than 40 years with very little loss in viability in cryostorage. Once species are in cryostorage, there's very little maintenance associated with the collection, typically just filling the dewars with liquid nitrogen. It's also quite space efficient, and a wide range of germplasm can be cryopreserved, including seeds, seed axes and embryos, tissue culture material, including shoot tips and callus cultures, and single cell cultures, fern spores, and pollen. Cryopreservation should be used to supplement and complement conventional germplasm storage methods, but can be used as a priority storage method when these conventional methods are inadequate, such as the case for recalcitrant seeded species, which can't be stored in a conventional seed bank. It can be the preferred method of germplasm storage for critically endangered plants, especially when there's very small quantities of material available, or when a very long-term storage is desired for the critical base collection. Cryopreservation started in Australia in 1992 with the successful cryopreservation of Grevillea scapidra shoot tips. This is a threatened species in Western Australia and was cryopreserved using a slow cooling method with the addition of DMSO as a cryoprotective agent. These cryoprotective agents are important for mitigating any ice damage that can occur during the cooling down to liquid nitrogen temperatures. After this point, in 1993, a wide range of native species, orthodox seed, were tested through cryopreservation. Of these 90 species tested, 68 species showed very good germination after cryopreservation. And this can be important for many of Western Australia's rare and threatened species that are amenable to cryopreservation using these seed. After this point, up till now, quite a variety of Australian germplasm has been cryopreserved, not only orthodox seed, but intermediate seed, such as water lilies. A wide range of orchid seed has been cryopreserved, as well as their associated mycorrhizal fungi. The protocorms have also been cryopreserved from Australian species, as well as somatic embryos from tissue culture. In total, 145 Australian species have been successfully cryopreserved, and as stated, a wide range of germplasm has been used to cryopreserve these species from seed axes, spores, protocorms, mycorrhizal fungi, callus tissue, as well as shoot tips, of which 41 of these species were cryopreserved using shoot tips. So why is this important? So Kings Park currently houses around 50 species in tissue culture. Many of these are rare and threatened, and cryopreservation is the only long-term storage option for these collections. Tissue culture is a good source of germplasm as it can supply a large amount of contamination-free shoots for experimental purposes, such as developing new protocols. And typically, the shoot tip will be used from these tissue culture shoots. So shoot tips make a good germplasm source for cryopreservation. One of the main advantages is they are quite small, so you get very good diffusion of the cryoprotective agents into the shoot tips, as well as good cooling and rewarming rates. In addition, shoot tips are good because they produce true to type plantlets. So you can avoid any somoclonal variation that can occur from callus cultures. So there's a range of cryopreservation methods for conserving shoot tips using cryo. One of the main ones is encapsulation dehydration. These samples are encapsulated in an alginate bead and then desiccated under a sterile airflow. Once sufficient water has been removed from the cells that the cytoplasm will naturally vitrify, the samples can be placed into cryostorage. However, it's only applicable to more desiccation-tolerant species. An alternative is the use of cryoprotective agents in the vitrification method. So these samples are placed into a solution of various cryoprotective agents called a vitrification solution, and these help desiccate the cells as well as promote vitrification of the cytoplasm. This is applicable to a wider range of species. However, many of these vitrification solutions can be toxic, so its use can be limited. So the current method most commonly used at Kings Park for conservation is the droplet vitrification method. So we take our tissue culture jars, excise out the shoot tip, and then pre-culture them on a desiccation media. This will help remove the majority of free water that can freeze. They are then incubated in a range of cryoprotective agents. So these vitrification solutions help bind up the remaining water and promote vitrification. For a droplet vitrification method, they are then transferred onto an aluminium foil strip, and the shoot tips are placed into small droplets of the vitrification solution. This helps improve the cooling and rewarming rates, as there's less vitrification solution that needs to cool and rewarm. At this point in time, they are plunged into liquid nitrogen and are considered cryopreserved. And then when required, the samples can be rewarmed as quickly as possible to avoid recrystallization of any water molecules, and then placed into a washing solution to remove any remaining vitrification solution and the cryoprotectants that can be toxic. After that point in time, they're placed onto recovery media to help promote the regeneration of the shoot tips. And this is typically done under no light to avoid any photooxidative stress, and then are measured and observed for regrowth and recorded. In addition, we quite often do a treated control, looking at how toxic the cryoprotective agents are on the shoot tips. So a alternative method might be required if the shoot tips show a lot of sensitivity to the cryoprotective agents. We have a few new techniques that we've been testing for the main cryopreservation collection at Kings Park. One of these is the vacuum infiltration vitrification method. So during incubation in the cryoprotective solutions, the samples are placed under a vacuum, which helps draw any air bubbles that might be trapped around the shoot tips and promote a more even diffusion of the cryoprotective agents into the shoot tips. This can also reduce the time the shoot tips need to be incubated in these toxic cryoprotective solutions and help promote better regrowth afterwards. Another is the cryomesh technique. So this method helps alleviate some of the physical handling of the shoot tips. So the shoot tips are placed onto a mesh strip and coated in alginate and stuck to that strip. They can then be transferred through all the different stages without handling each of the individual shoot tips and potentially damaging them during the process, but still maintains a very high cooling and rewarming rates of the droplet vitrification method. And more recently, we've been testing some flash drying. So this uses very high airflow rates over the samples, typically with compressed air, to rapidly dehydrate the shoot tips. So this method avoids the use of these toxic cryoprotective agents, but relies on a bit more of a desiccation tolerant species. However, if they spend very little time at the damaging low moisture contents, more samples could be cryopreserved using this method. So we currently have an honor student working on this, and Typically in a laminar flow, you'll be looking at about 5-6 hours of drying before the samples are sufficiently desiccated. While this method, we're seeing the samples sufficiently desiccated after about 2.5 hours. So really reducing the time it takes to get down to the optimal moisture content in the cells. However, there's always issues and difficult species with cryopreservation, and not all species are amenable to cryopreservation. We've observed quite a few things trying to cryopreserve our Australian species. A common one is very slow growth post-cryopreservation, with many species taking months to start regrowing and regenerating into a normal culture. We also see a fair bit of abnormal growth, as well as quite a few species are sensitive to the toxic cryoprotectants and will require some alterations to the standard protocols to try and alleviate the stress. We've also seen quite a few species show a lot of oxidative damage after the cryopreservation procedure, where the shoot tips will just go completely black. And work has been done looking at oxidative damage during the individual stages of cryopreservation and what can be done to alleviate some of this damage. So a lot of work to be done to understand how the Australian species respond to cryopreservation so we can apply it to a wider range of species. So the current shoot tip collection in cryo storage at Kings Park is 37 species currently in our dewars. There's 117 clonal accessions, capturing some of the diversity of these species, and about 7,700 actual shoot tips in our dewar. So 36 species, Australian species, have a published protocol for how to cryopreserve them using tissue culture material. And the oldest record we have still in our dewars is from 1999. It'll be great to pull it up and see if it's still viable after 20 years. And this is just our shoot tip collection. The Kings Park cryopreservation collection also includes seeds and mycorrhizal fungi associated with some of the orchid seed we have in storage. So I'd just like to thank my supervisors, Ricardo Mancera and Eric Bunn, and all the staff and students at Kings Park Science, as well as the co-authors on our germplasm guidelines cryopreservation chapter. So please check it out, and thank you for your attention.
Hello, good afternoon all. Today I'll be talking on extracting and cryopreserving embryonic axis of recalcitrant seeds. I'll be focusing more on the challenges and the possible preventive measures to overcome them. I will give examples from our previous work and will finish it off with our current work on Syzygium myri, a New Zealand recalcitrant Mataceae species. We know that for cryopreservation of recalcitrant seeds, we need to work with the embryos or embryonic axis. The success of cryopreservation can be limited by physical damage to the embryo during isolation, inter- or intra-population tissue heterogeneity, which could be physical or physiological differences, variable responses to different drying techniques and rates, oxidative stress associated to excision, drying, and cryopreservation procedures. If we are using cryoprotectant, uniform permeation is critical, and this could affected by size, lipid fluidity, viscoelasticity, and hydrophobicity of the tissue. As zygotic embryos have complex tissue structure covering radical and plumal meristem, they will respond differently with cooling and thawing procedures. Lack of knowledge in vitro growing of the tissues also could affect the success of cryopreservation. As I mentioned before, zygotic embryos have complex tissue structure, and the first challenge would be to locate the embryo inside a seed and extracting it without damage. The excision technique will vary from endospermic and cotyledonary embryo. For endospermic embryo, there usually will be a cavity between the endosperm and the embryo, and with the correct method, we can scoop out the embryo. But with the cotyledonary embryo, it always will involve a surgical procedure to detach the embryo from the cotyledons. For seeds with larger embryos, we need to work on their embryonic axis. Embryonic axis may require different in vitro growth medium compared to the full embryos. One study we conducted with cycad showed that the axis having at least 10% less germination compared to full embryos for the 2 of the 3 species we studied. Also, bear in mind that the axis would require oxygen supplement at the latest stage for root initiation and development. Collecting the seeds at the correct maturity state is critical to achieve optimum survival following desiccation and cryopreservation. We tested 3 different maturity states for Sterculia cordata, a tropical recalcitrant species. Seed development stage 3 or SDS3 was when the seeds are fully matured with red fruits and black seeds. SDS2 was a week before full maturity, and SDS1 was 2 weeks before full maturity. As you can see from the graph here, the immature embryos are more sensitive to desiccation and cryopreservation. Embryos collected at full maturity are more tolerant to desiccation and therefore amenable for cryopreservation. Embryo oxidation and phenolic compound production is a widely reported observation in recalcitrant seeds during excision and drying. The severity of oxidative stress varies between species and families. Here I have an example of 2 palm embryos which show oxidation or browning immediately after excision. We tried various antioxidant incorporation in the growth medium, but once the embryos show browning, viability was compromised. We finally optimized the technique by imbibing the embryos in water for 24 hours immediately after excision and used these embryos for cryopreservation. As you can see here, we eliminated the embryo browning in both species. Another technique we tried was to have some endospermic tissue to cover the embryo and either to keep or remove the operculum. This method was also successful, but germination was delayed compared to the fully excised embryos. When using cryoprotectants, we want to make sure the permeation is fast and uniform. However, this is influenced by many factors such as concentration of the cryoprotectant, exposure time, temperature, permeability, and diffusion pathway length. To overcome this complex problem, we applied cryoprotectant at vacuum for 3 species with varying morphology, physiology, and lipid chemistry. These are papaya, passion fruit, and bay seed embryos. As you can see here, they all are oily and have different fatty acid compositions. And these are the results. We managed to achieve higher regrowth using vacuum infiltration vitrification. The exposure time under vacuum was significantly reduced. The regrowth was improved from 25% to 55% in papaya and from 55% to 70% in bay. We believe that the vacuum infiltration vitrification could assist in fast and uniform permeation of cryoprotectants. What do you get when you put all the problems of recalcitrant seeds together? You get Syzygium myri. Yes, Syzygium myri or swamp myri is a New Zealand recalcitrant Myrtaceae species that we are currently working on. With the incursion of myrtle rust in New Zealand, we are desperately developing cryopreservation protocol for conserving this species. And I can list all the problems associated with Syzygium myri here. The seed biology is not fully understood. We have a very narrow window of seed availability to develop the protocol, and the seed maturity greatly vary with and within populations. There are inter and intra-population differences in response to desiccation. The embryos are too large for cryopreservation, therefore we have to work with the excess. Oxidative damage to embryos during excision and desiccation is quite big. There's no optimized protocol for embryo sterilization, and uneven drying of embryos prior to cryopreservation, therefore affecting survival. Non-uniform permeation of cryoprotectants, no prior information on cryopreservation method, and lack of information on in vitro growth medium and condition poses more problem to this species conservation. Just to give you some idea on the inconsistency in embryo maturity state at collection, you can expect to see the fruits at different physiological maturity at one time. Embryos collected from different populations at different years also showed different responses to drying. We used a DSC to see the crystallization events of individual embryos with different sizes. We noted that the onset, peak and end temperatures could vary by 3 degrees centigrade. The peak area is directly relevant to the embryo size. To avoid these differences, it is recommended to have uniform samples where possible. Have more replications to reduce variation between samples. For DAC measurements, normalize outputs by sample weights. As you can see here, the enthalpy after being normalized by sample weight does not show the same trend as the area. Drying at a control environment can eliminate the differences between samples. And finally, using rapid drying instead of slow drying can also assist in uniform desiccation. Here, I would like to share with you a video on Cygesium myri embryo excision and initiation in vitro. As the embryo oxidation rate was quite high, we modified the protocol by having the seeds submerged in ascorbic solution. Embryo excision was carried out under ascorbic acid solution. Excised embryos were kept in ascorbic acid solution. All pre-culture, encapsulation, vitrification, and regeneration media were incorporated with ascorbic acid. Following above modification, we managed to retain green embryos following encapsulation and pre-culture. This slide summarizes our current work on Sergesium myri. We are optimizing cryopreservation protocol using encapsulation dehydration, vitrification, and cryoplates techniques. We are using a DSC to understand the biophysics. We are using biochemical stress markers to elucidate and prevent oxidative stress. We are also testing other materials such as plumule and sheet tips for cryopreservation, just in case if embryo cryopreservation is not achievable. And with that note, I would like to end my presentation by thanking the Fenner Conference Organizing Committee for giving me this opportunity. Thank you. And there are various people and organizations to acknowledge. And thank you all for your attention. Thank you.
Hello, my name is Daniel Ballesteros and I'm researcher at Royal Botanic Gardens Kew. I'm going to share with you some thoughts and practical considerations for the development of furnace spore ex situ collections, hoping that this talk inspire the creation of at least one new furnace spore bank within Australia. Pteridophytes, to which I will refer as ferns in this presentation, is a paraphyletic group of plants that includes the lycophytes, such as club mosses, spig mosses, caworts, and the monilophytes, which include the true ferns, whisk ferns, and horsetails. Differently than seeded plants, they reproduce by spores and lack flowers and seeds. The main life form we see from the ferns are the sporophytes. These disperse by haploid spores, and after germination, they develop a small and independent gametophyte. Gametophytes will grow and mature and sexually reproduce, finishing this process in the development of a new sporophyte. "155 species of ferns, which represents 16% of the species evaluated, are threatened with extinction globally. In addition, 55 species have been evaluated as near threatened at global scale. At global scale, priority conservation areas include tropical and subtropical montane forests due to the high endemism and human pressure. But ferns are also included in other regional and local programs of conservation prioritization, as we can see in the example extracted from the European Red List of Ferns. Analogous to seed banks, furnace spore banks are the preferred ex situ conservation tool, as high genetic diversity can be preserved in a small space with minimal technical requirements. However, to our knowledge, only about 10 spore ex situ collections are active globally, which represent a small number compared to the over 1,500 seed banks known. Guides for furnace spore conservation have not been frequently available in the literature due mainly to the lack of consensus in the storage conditions to be used, which was related to a poor knowledge of their storage behavior, the low number of researchers working on the topic, and the low funding invested on fern conservation compared to other plant groups. However, in the recent years, and based on the scientific knowledge acquired in the last 15 years, diverse furnace spores conservation guidelines are available in English. From a preservation point of view, one must know that there are two types of furnace spores: chlorophyllose or furnace spores that age very fast due to their high chlorophyll content, like willow seeds, and non-chlorophyllose or non-green spores, which retain viability for longer. Due to the short lifespan of green spores, they should be processed very fast to retain the maximum viability. Spores, like seeds, must always be collected at maturity, as in nature spores or spores left in open sporangia from previous seasons won't tolerate drying or will show short lifespan respectively. Mature spores are found in fully developed and closed sporangia. The identification of mature sporangia can vary among species, but generally they will be found on the youngest but fully developed healthy leaves. Mature sporangia are close and are bright in color, ranging from yellows to dark brown for non-chlorophyllous spores. For species with sori covered by indusia, which is this structure over here, the color of this structure turning from green to yellow or brownish helps to identify the mature sporangia. On the other hand, sporangia in ferns with chlorophyllous spores tend to dark green when spores are mature and become brown after spore release. Slightly different ways exist in the literature to assess the maturity of spores in fern species with sporangia within strobili, like horsetails, or other structures, like the sporocarps in Marsilea. For fern spore collection, generally, fronds or strobili are dried over paper in the lab for a few hours or days. When sporangia open, the spores fall on the paper and are easily detected. It is recommended to sieve spores to eliminate frond residues, as a clean collection is less likely to become contaminated during propagation. Spores are desiccation tolerant, like orthodox seeds, but suffer if ultra-dried below 10% relative humidity. Drying conditions recommended vary slightly depending on the final storage temperature. These variations are suggested to ensure that the moisture at storage is between 15 and 25% of the equilibrium relative humidity, the moisture conditions that allow the longest lifespan. Like orthodox seeds, spores can be stored in the fridge or freezer at temperatures between 5 and -20 degrees Celsius. But to ensure their long-term preservation, it is recommended to store them at cryogenic conditions. Freeze-thaw cycles must be avoided, so it is always good to have subsamples of the same spore accession. But fern spore banks, in addition to their use in conservation, have a great value for fundamental research that could benefit the improvement of other gene banks such as seed banks. For example, green and non-green spores have been used as unicellular model to study the fundamental mechanisms that control aging in seeds with short lifespan, which are related to the chlorophyllous and oily seed types. For example, we have shown how global mobility of the lipid domain during storage at sub-zero temperatures is apparent by the higher enthalpy of melting events in store compared with fresh fern spores. However, the mobility of the storage lipids during liquid nitrogen storage is considerably reduced compared with counterparts stored at -20 or -80 degrees Celsius, which explains the extended longevity when these systems are cryopreserved. At -20 degrees Celsius, global motion of the storage lipids is very apparent by the increase of enthalpy of melting and can lead to the anomalous low rapid aging detected in some systems. These results have helped us to propose an aging mechanism for oily seeds, in which, when exposed to sub-zero temperatures, lipids slowly crystallize and compact their volume within the glassy matrix of the stored seed. We have hypothesized that over storage time, the forces of compression or the voids created after the shrinkage of the lipids increase the chance for oxidative stress or structural collapse, promoting in that way the anomalous fast deterioration detected in some oily seeds. Another example are the chlorophyllous seeds of willows and poplars, known to be desiccation tolerant but very short-lived in the dry state. Their fast aging has been related to photooxidative stress induced by an uncoupled photosystem in the absence of water. However, it is not clear why chlorophyll seeds age also fast in the dark state. We have found that antioxidants differ among chlorophyllose spores with differing longevity, with, for example, plastochromanol 8, a type of tocochromanol involved in protection of chloroplast membranes, being found only in the longest-lived green spores. These antioxidants regulate the spore lifespan in the dry state by preserving the photosynthetic activity in both light and dark conditions, being aging related to the loss of antioxidant capacity over the storage time. Recently, we have performed experiments in the UK National Neutron Source Facility, which is a kind of cyclic particle accelerator. In our experiments, we used dry furnace spores from 3 different species. In all species, molecular mobility was detected at temperatures as low as -223 degrees Celsius. All 3 species showed 4 distinct dynamic regimes, which were mainly related to molecular vibrations at the lower temperature range and to anharmonic motions of 2 distinctive nature, such as side chain rotations and molecular bendings. These dynamic regimes showed differences between species that could be related to their chemical composition and storage stability. This is the end of my presentation, and as a take-home message, I would like to highlight 3 basic points. First, that furnace spore banks can provide a high-quality germ plants not only for ex situ conservation of ferns, but also for fundamental research that can benefit seed science and conservation. Furnace spores tolerate similar drying to orthodox seeds and could be curated and stored at the standard conditions of seed banks. However, due to the short lifespans of chlorophyllose spores at all storage temperatures and some non-chlorophyllose spores at -20 degrees Celsius, fast processing and cryogenic storage is recommended for their long-term conservation. Thank you very much for your attention, and I hope you liked the presentation.
Thank you to Dr. Pence and all our Session 2 presenters. Valerie and Daniel are not able to join us live today due to time zone differences, but they are very happy to answer questions by email. I'd like to welcome Jayanthi and Bryn now, and thank you for making time to answer questions this afternoon. Welcome, Bryn. Welcome, Jayanthi.
Thank
So
you.
we have some great questions from the audience today, and I'll start with one for both of you. So Bryn, if you go first, what are the basic requirements and costs associated with cryo storage?
I think it's a bit dependent on what you're planning to cryo store. So if you're working with orthodox seed, you literally need a dewar and a constant supply of liquid nitrogen. But as soon as you start working with more exceptional species and you're going into tissue culture, you need all the facilities associated with tissue culture, and that becomes a lot more costly and a lot more setup involved. So I think it's very dependent on your application on how you want to cryopreserve something. But for something like orthodox seed, you don't need a particularly large dewar for experimental purposes, and can expand with new dewars as your collection grows from that point in time.
Oh, fantastic. And Jayanthi, do you have anything to add there?
Yeah, I agree with Brian. So the setup cost is usually will be high. And it will be a one-off cost. It depends on your purpose of storage and how large scale you're going to go with. It can start anything from 20,000 to 100,000 to set up. And then the maintenance cost is, again, depends on the material you're storing. Seeds really require minimal supervision and manipulation before storage. And you don't need a lot of in vitro work before storage. But if you're working on clonal materials, that will be more expensive.
Yeah, fantastic. Thanks, Jayanthi. Another question for you, Bryn. Have you found plants from drier habitats any easier to cryopreserve than plants from wetter habitats?
Yes, generally, but not always the case. So some plants grow very differently in tissue culture than what they look like in the wild. And there's a lot of water available to the plants in tissue culture, so they can actually have a much higher water content compared to what they will normally grow at. So some species do tolerate desiccation a lot better once you start removing water from them. And we found some of our grevillea species very tolerant to desiccation during cryopreservation. And the same, we're working on some of the Syzygium species, and they come from rainforests and they're very sensitive to that. So there is a little bit of similarity there, but there is always exceptions where we have very difficult species that you would think would be easy and have been very difficult to cryopreserve.
Yeah, fantastic. And from a question from Vitri. Vitri asks, is there a best material to use in cryopreservation if there's normal growth and they're highly viable? Where would you start?
I think you would always start with seed. So if you got orthodox seed, they're by far the easiest to cryopreserve. You just need to be dried down, placed into liquid nitrogen. After that point in time, some of the intermediate species could really benefit. They don't require much more processing, but they might benefit from the cooler temperatures and extend their lifespan. And then, yeah, I think tissue culture material really becomes sort of last resort, but there is reasons for using tissue culture material, especially for rare and threatened species, to capture as much diversity as possible.
Fantastic, thank you. And how, Jayanthi, this could be for you as well. How do you prioritise which species you intend to cryopreserve in the future?
I will start with that. It all depends on, I think we follow Valerie Pence's talk, and we know that we need to prioritize based on how much threatened they are, the rarity of the species, and the nature of threats. And for example, all the Metaceous species that's known to be non-threatened in the last 2 or 3 years ago now seem to be threatened even in New Zealand, and same with Australia. So the priority of the conservation depends on the threats of the species in the nature, how vulnerable they are in the nature. And maybe Bryn can add more on that.
Yeah, I think our cryopreservation is focused really on rare and threatened species and what we have in the tissue culture lab. So what we've been able to collect really determines what we've cryopreserved at this stage. We would like to have a more focused approach and have a list and try and work our way through it. But it is sometimes down to us to what we've been able to collect and initiate into tissue culture.
Yeah, I understand that. And I think there's a great table in the germplasm guidelines that lists what's already cryopreserved in Australian, in collections at Plant Bank and at Kings Park as well, which is fantastic. Yeah. I'll just acknowledge now that there are some great questions and comments about banking of fern spores in the chat. I think we might pick that up this afternoon in our panel session because, there's a lot of interest and also developing resources about that. There's a couple of quite detailed questions here for you, Bryn. One from Dave Merritt. In desiccation tolerant but freezing sensitive seeds, we often only see the damage from freezing manifest after months in sub-zero temperature storage. Is that something you notice with shoot tips as well? Do you need some time in storage before the damage is evident?
I think shoot tips are a little different because we always add cryoprotective agents to them, which really helps stabilize the glass and its stability under liquid nitrogen temperatures. It is something that's certainly be seen in embryonic axes and short-lived seeds under cryo storage. But I think shoot tips are quite a different kind of tissue, and the addition of cryoprotective agents can really help stabilize the sample under liquid nitrogen temperatures. So it's something that's not commonly seen with shoot tips, where the viability is normally very good or very similar to what it was initially in the long-term trials that have been done. We haven't done any particular long-term trials in our cryo storage. So it's something we want to do in the future and pull out some of our old collections and see how viability is in those old collections. So be interesting to see if we do find something after 20 years or somewhere around there.
Yeah, it's fantastic. Yeah, that will be really interesting to follow as you get them out and test them again. We also have a question in the chat from Aulia. Thanks for your question, Aulia. It's on a species of Dipterocarpaceae, Vatica venulosa. Now, she's tried lots of methods, plant vitrification solution 1, 2, and 3, desiccation for 0, 5, and 10 hours, a range of moisture contents, a range of thawing protocols, growing it again on woody plant medium. Still no embryo successfully grown. I'm sure both Jayanthi and Bryn empathize with you, Aulia. Where would you go when you've run up against so many walls and you're not having any success?
Well, I can start with that. Our experience with swarm Murray is a similar experience, and I have experience also working with other Dipterocarpaceae species. From my talk, you also can see the accumulation of stress. It will start at the very beginning when you extract the embryo. So you have to work the protocol step by step to optimize each protocol. So start with the extraction of the embryos, optimize that protocol first, make sure that you get really healthy embryos, and then get on the next step, where is the desiccation. So sometimes the embryos can be sterilized beautifully, but then when you move on to the desiccation protocol, you're going to have problem, either is a desiccation sensitivity or oxidative stress. And if you optimize that protocol and then move on to the next one, is either adding cryoprotectants or drying the embryos to optimum moisture content for cryopreservation. And then the next step is freezing, thawing, and then subsequent recovery. So optimize protocol at each level and test for regeneration at each level as well, so that you know where the problem occurs. And if the problem occurs at the very beginning stage, then you need to rectify that problem at the very beginning step itself, because the stress can accumulate. And if you have this stress starting at the beginning, at the end of your protocol, you will have 100% stress and no recovery.
Yeah. Do you have anything to add there, Bryn?
No, I agree. If your samples are starting to struggle right at the start, the added stresses of cryopreservation can really reduce any likelihood of you getting it survival at the other end. So it's very important to take samples at each stage, after each stage of cryopreservation, and assess how well the plants are going to grow after that stage. And it can help identify the most stressful stages and where you can start focusing on alternatives to that stage and what you can start doing to improve survival up to that stage, which will help improve the overall process.
Fantastic. Thanks, Bryn. So hang in there, Aulia, and certainly draw on the network of speakers who've presented today. I know they're all open to problem solving and questions via email down the track. Thank you. We'll finish up with one last question. It's from Diane, and I think it's really pertinent. These types of ex situ conservation can be quite resource hungry, and Diane is asking, do you have any advice for a management strategy for establishing and running cryostorage facilities under limited funding and other limited resources?
Okay, I'll have a go. It is very difficult with limited funding, especially if you're in the tissue culture side, where the maintenance of the tissue culture collection can be pretty much most of your time goes into the maintenance. So I'd say a very clear goal of prioritizing species into cryopreservation, and a bit of something we sort of trying to focus on now, on trying to clear out our tissue culture collection and store them in cryopreservation. It frees up a lot of time to work on new species, if you're not maintaining double collections, both in cryostorage and tissue culture. So prioritizing the time you have to work on cryopreservation and trying to get species in there at sufficient levels that you can then remove them from the tissue culture environment can help free up a lot of your time for working on new stuff. So it's a suggestion I have from what we're trying to do at the moment.
Yeah, fantastic. Thanks, Bryn. Jayanthi?
Yeah, it's always a decision-making thing. Prioritizing is important, as well as the economic impact is also important. So if you're making a decision at higher level, whether you're going to use cryopreservation or tissue culture collection or living collection, what is the purpose of collection? Why are you conserving this? Is it for long-term or is it for short-term use? It's quite important. And for cryopreservation, we always can convince, like, though the setup cost is quite high, but the long-term benefit is really critical and it's really evident. So we can show that evidence and saying that, yes, you can use cryopreservation. Is initially high cost, but then once you have established and you have the skill to do it, it's really reliable way of conserving, especially for threatened and recalcitrant species. There's no other way of doing it.
Yeah, fantastic. I did say that was the last question, but I've got one more for you, Jayanthi. A question from Marie. Do you know of any fern spore banks in New Zealand, given the environmental threats to the native ferns and their importance to the Maori?
I'm not aware of any spore banks in New Zealand, unfortunately, but this is something I need to investigate myself. So thanks, Marie, for that question.
Thanks, Marie. And we will pick up on some of those questions about ferns in this afternoon's discussion. Okay, please stay online to answer 2 new poll questions before you log off from session 2 today. You'll need to use the new Zoom link provided by Aranex to join session 3, this afternoon's panel discussion, just before 2:15. Thank you so much to Bryn and Jayanthi.